natural reactors to the complex, marvelous tapestry of invention and ingenuity that we see around us? The answer, of course, is unknown, but there are clues that derive from the properties of life itself, and in particular from an inner core of deeply conserved reactions common to almost all life on Earth today. This core of metabolism, a living inner fossil, preserves echoes of the most distant past, echoes that are consonant with primordial origins in an alkaline hydrothermal vent. There are two ways of approaching the origin of life, the bottom-up and the top-down. So far, in this chapter, we have taken the bottom-up approach, considering the geochemical conditions and thermodynamic gradients that most likely existed on the early Earth. We have come up with warm, deep-sea hydrothermal vents, bubbling hydrogen gas into an ocean saturated with carbon dioxide as the most likely setting for the origin of life. Natural electrochemical reactors would have been capable of generating both organic molecules and energy, but we haven't considered yet exactly what reactions are likely to have taken place or how they led to life as we know it. The only true guide to how life came to be is life as we know it today, that is to say, the top-down approach. We can catalogue the properties shared by all living things to reconstruct the hypothetical properties of a last universal common ancestor, known fondly as Luca. So, for example, because only a small subset of bacteria is capable of photosynthesis, it is unlikely that Luca herself was photosynthetic. If she were, then the great majority of her descendants must have abandoned a valuable skill which seems at best improbable, even if it can't be excluded with certainty. Conversely, all life on Earth shares common properties. All living things are composed of cells, excluding viruses which can only operate within cells. All have genes made of DNA. All encode proteins by way of a universal code for particular amino acids. And all living things share a common energy currency known as ATP, adenosine triphosphate, which functions as a sort of 10-pound note, capable of paying for all kinds of work about the cell. More on this later. We can infer reasonably that all living organisms inherited their shared properties from that remote common ancestor, Luca. All life today also shares a common core of metabolic reactions, at the heart of which is a little cycle of reactions known as the Krebs cycle, after Sir Hans Krebs, the German Nobel laureate, who first elucidated the cycle in Sheffield in the 1930s after fleeing the Nazis. The Krebs cycle occupies hallowed ground in biochemistry, but for generations of students it felt like the worst kind of dusty ancient history to be rote-learned in time for exams and then forgotten. Yet there is something iconic about the Krebs cycle. Pinned to the walls of cluttered offices in biochemistry departments, the kind of office where you'll find piles of books and papers on the desk, spilling over onto the floor and into the bin, unemptied for a decade, you'll often find a faded, curling, dog-eared metabolic chart. You peer at it with a mixture of fascination and horror while you wait for the professor to return. They are shocking in their complexity, like a lunatic's version of an underground tube map, with little arrows running in all directions, looping back round on each other. Although faded, you can just make out that these arrows are all colour-coded for different pathways, proteins in red, lipids in green, and so on. Down towards the bottom, somehow giving the impression that it is at the centre of this insurrection of arrows, is a tight little circle, maybe the only circle, indeed the only ordered bit on the whole map. That's it, the Krebs cycle. And as you peer at it, you begin to appreciate that virtually all the other arrows on the map somehow spin off from the Krebs cycle, like the spokes of a mangled wheel. It is the centre of everything, the metabolic core of the cell. The Krebs cycle doesn't feel so dusty anymore. Recent medical research has shown it to stand at the heart of the cell's physiology as well as its biochemistry. Changes in the rate at which the cycle spins affect everything from aging to cancer to energy status. But what came as an even bigger surprise is that the Krebs cycle can go backwards. Normally, the cycle consumes organic molecules from food and spins off hydrogen, destined for burning with oxygen in respiration, and carbon dioxide. The cycle therefore not only provides precursors for metabolic pathways, it also serves up the little packets of hydrogen needed for generating energy as ATP. In reverse, the Krebs cycle does the opposite. It sucks in carbon dioxide and hydrogen to form new organic molecules, all the basic building blocks of life. And instead of releasing energy as it spins, the reverse cycle consumes ATP. Provide it with ATP, carbon dioxide and hydrogen, and the cycle spins out the basic building blocks of life, as if by magic. This reverse spinning of the Krebs cycle is not widespread, even in bacteria, but it is relatively common in the bacteria that live in hydrothermal vents. It is plainly an important, if primitive, way of converting carbon dioxide into the building blocks of life. The pioneering Yale biochemist Harold Morowitz, now at the Krasnow Institute for Advanced Study, Fairfax, Virginia, has been teasing out the properties of the reverse Krebs cycle for some years. In broad terms, his conclusion is that given sufficient concentrations of all the ingredients, the cycle will spin on its own. It is bucket chemistry. If the concentration of one intermediate builds up, it will tend to convert into the next intermediate in succession. Of all possible organic molecules, those of the Krebs cycle are the most stable and so the most likely to form. In other words, the Krebs cycle was not invented by genes. It is a matter of probabilistic chemistry and thermodynamics. When genes evolved later on, they conducted a score that already existed, just as the conductor of an orchestra is responsible for the interpretation, the tempo and the subtleties, but not the music itself. The music was there all along, the music of the spheres. Once the Krebs cycle was spinning and provided with a source of energy, side reactions would have been almost inevitable, giving rise to more complex precursors such as amino acids and nucleotides. How much of the core metabolism of life on Earth arises spontaneously and how much is a later product of genes and proteins is an interesting question and one that is beyond the scope of a book like this. But I would like to make one general point. The great majority of attempts to synthesize the building blocks of life have been too purist. They start with simple molecules like cyanide, which have nothing to do with the chemistry of life as we know it. In fact, they are anathema to it. Then attempt to synthesize the building blocks of life by playing around with factors like the pressure, temperature, or electrical discharges, totally unbiological parameters, all of them. But what happens when you start out with the molecules of the Krebs cycle and some ATP, ideally in an electrochemical reactor like that proposed by Mike Russell? Just how much of our dog-eared metabolic chart arises spontaneously from these ingredients in a kind of ethereal cast that gradually fills up from the bottom with the thermodynamically most likely molecules? I am not alone in suspecting quite a lot of it, perhaps up to the level of small proteins, strictly polypeptides, and RNA, at which point natural selection begins to take over. All this is a matter of experimentation, and these experiments are mostly yet to be done. For any of it to be realistic, we need a nice steady production of that magic ingredient ATP, and on that score you may well feel that we're getting ahead of ourselves, trying to run before we've learnt to walk. How do we generate ATP? The answer I find most persuasive comes from the brilliant, if frequently outspoken American biochemist Bill Martin, who forsook the US to take a job as professor of botany at the University of Dusseldorf. From here, Martin has been the source of a steady flow of iconoclastic ideas about the origin of almost everything that matters in biology. Some of it may be wrong, but he is always thrilling and nearly always makes one see biology from a different point of view. A few years ago, Martin sat down with Mike Russell, and the pair of them attacked the transition from geochemistry to biochemistry. Since then, the insights have been flowing free. Let's take the ride. Martin and Russell went back to basics, the flow of carbon into the organic world. Today, they note, there are only 5 metabolic pathways by which plants and bacteria incorporate hydrogen and carbon dioxide into the living world to generate organic matter, one of which is the reverse Krebs cycle, as we've seen. 4 of the 5 pathways consume ATP, as does the Krebs cycle, and so can only take place with an input of energy. Yet the 5th pathway, the straight reaction of hydrogen with carbon dioxide, not only produces organic molecules, but it also releases energy. 2 groups of ancient organisms do exactly this via a series of broadly similar steps, and one of these 2 groups we've already met, the archaea, that thrive in the lost city vent field. If Martin and Russell are right, the remote ancestors of these archaea were performing the same set of reactions in an almost identical environment 4,000 million years ago at the dawn of life. But the reaction of hydrogen with carbon dioxide is not quite as straightforward as it sounds, for the 2 molecules don't react spontaneously. They are rather shy and need to be persuaded to dance by a catalyst. And